Hello student, welcome to EPG Pathsala. I am Dr. Ajit Jaiswal from Department of Anthropology, Pondicherry Central University, Puducherry. Today we are going to discuss about a modules called as genetic drift and its consequences under paper human population genetics. In this particular modules we are going to discuss about what is genetic drift and other feature related to genetic drift or the feature in the form of concept related to founder effect, concept related to bottleneck effect. So, before going to discuss about the genetic drift, bottleneck effect or founder effect, it is important for us to know the elementary information or initial information regarding what is genetic drift, how we explain what is the historical feature. So, genetic drift is a form of evolution that does not usually result in adaptation. It is usually said to be evolution of a neutrally adaptive trait. In other way, different alleles may increase or decrease with respect to each other, but the changes do not represent adaptation. Genetic drift always a second mechanism for evolution and proposed by civil rights in the year 1930s. The mechanism of genetic drift is a chance associated, chance factor is there with recombination in small population, chance small population. When a small group separates from a larger group or reproduce only among themselves, allele frequency may change as a result of chance sampling from the whole. This change in allele frequency that occur when a small group separate from the large whole is termed genetic drift. It is like reaching into a bag of jelly beans and by chance grabbing only green and yellow one. The allele frequency changes in genetic drift are random and unpredictable. So, chance, random, small population and unpredictable. Genetic drift. When genetic drift occur, as we already told you that, in fact we have already discussed that drift is a random variation in allele frequency from one generation to the next. Random sampling, the important condition is that random sampling is required from the parental population to form the next generation. It is caused by Mendelian segregation and finite population size. In large population, it produces very slow changes compared to selection. However, over a long period of time, drift has a large cumulative effect on allele frequency. In a small population, isolated population produces rapid changes. So, genetic drift occur when the population size plummet due either to migration to a natural disaster or geographic barrier that isolates a small pocket of people or to the consequences of human behavior. Members of a small community might reproduce only among themselves which keep genetic variation within the ethnic group. Genetic drift is also seen within large cities. Pittsburgh, Pennsylvania and New York City are more mosaic of groups with distinct ethnic flavor than melting pots of mixed heritage. Some group of people become isolated in several ways, geographically, linguistically and by choice of partners. Such populations often have a high incidence of several otherwise rare inherited conditions. The native resident of the Basque countries in the western part of the Pyrenees mountain between France and Spain, for example, still speak remnant of Euskera, a language the first European settlers brought in more than 10,000 years ago. The Basque have usual frequency of certain ABO and Rh blood type, rare mitochondrial DNA sequence and self-surface antigen pattern and a high incidence of mild form of muscular dystrophy. Let's discuss about the case study. It shows the impact of genetic drift on the greater prairie chicken. Here in this case, loss of a prairie habitat is equal to reproduction of population or reduction of population. In a direct sense, we can say that the surviving bird had a low level of genetic variations and only 50% of there is hatched. So there is a loss of prairie habitat has a direct relation with the reduction of population. Factors for genetic drift. Our genetic drift has several important effects on evolution. First, let's discuss about the factors. The following factor must function for genetic drift to work, like allele involved must not hold significant selective advantage over each other. Second, the population involved must be quite small and sexually reproducing. Third, there can be no significant immigration or immigrations of one allele as compared to another. For many population and allele, these factors hold true. For example, many alternative allele of genes produce protein that are essentially equivalent in function. The differences in their amino acid structure result in no noticeable differences in function in the environment the organism is found in. In the recombination process, there are two events that can easily affect the frequency of allele in small population, such as reproductive division of meiosis and syngamy. As population gets smaller and smaller, it becomes increasingly likely that allele might change in their abundance in the offspring as compared to the parents. Genetic drift works the same way. One allele or the other increase simply at changes in small population. So we can say that genetic drift has several important effects on evolution. First one, drift reduce genetic variation in population, potentially reducing a population's ability to evolve in response to new selective pressure. Genetic drift acts faster and has more drastic result in small population. This effect is particularly important in rare and endangered species. Next important effect is genetic drift can contribute to speciation. For example, a small isolated population may diverge from the larger population through genetic drift. It has also some other important factor like effect of genetic drift can be very strong when compounded with many generations. So now let's try to discuss or highlight the main aspect of genetic drift. There are altogether four important aspects around which the concept of genetic drift revolves. First, the direction of genetic drift is unpredictable. In n generation, that's why I call this a chance factor. In n generation, the allele frequency may increase or decrease with equal probability. It may increase, it may decrease. So the chances is equal in both the direction. Second aspect is that the magnitude of genetic drift depend on the population size. If the population size is smaller, the smaller the population, the larger, or what you call the change from one generation to another. So if the population size is small, so the possibility of genetic drift or changes will be higher. Third aspect is that the long-term effect of genetic drift is to reduce genetic variation within a population. Population heterozygosity is reduced and allele are lost. And the final one, genetic drift causes population to diverge from one another. So ultimately lead to the divergence, moving away from the original one. Characteristics of genetic drift. Phenomena while share some characteristics with genetic drift such as founder effect and migrations are important in evolution. Let's first discuss about the founder effect. Founder effect is a related phenomena of Ernst-Mayr. Here the idea is that new populations that are found by relatively few individuals will be by the small founding genotype. Differences between them and other populations may have little to do with adaptation and instead simply to the result of chance. The founder effect is an excellent example of the potential role that history can have in the evolutionary change. Biased migration, where one genotype preferentially leave a population, might also in some cases by a factor. So, the founder effect occurs when a few individuals become isolated from a larger population, and here the allele frequencies in the small founded population can be different from those in the large parent population. Now, when we discuss about the concept of founder effect, let's try to find out the founder effect in human population. Founder effect is a related phenomena of Ernst Mayr. Here the idea is that new populations that are founded by relatively few individuals will be biased by the small founding genotype. Differences between them and other population may have little to do with adaptations and instead simply be the result of chance. The founder effect is an excellent example of the potential role that history can have in evolutionary changes. Biased migration, where one genotype preferentially leave a population, might also in some cases be by a factor. A common type of genetic drift in human population is the founder effect, which occur when a small group leave a home to found a new settlement. The new colony may have different allele frequency than the original population. Founder population can amplify certain allele. This shows up in increased disease frequency. Consider an isolated community of about 10,000 members of the Church of Jesus Christ of Latter-day Saint, who live on a plateau on the Utah, Arizona border. Half of all known cases of fumarate deficiency, which causes mental retardation, seizure and coma, occur in this area. Nearly 80% of the population descends from two original settlers. Widespread cousin marriage as well as polygamy, that is man with multiple wives, and social isolation have joined the powerful founder effect to keep the disease in the population. In 2008, the US government removed many children from this community on suspicious of abuse. Some of the children returned a year later, but some choose to remain in the outside world. A powerful founder effect appears in the French-Canadian population of Quebec. Their lack of diversity in disease-causing mutation reflect a long history of isolation. Considered breast cancer caused by BRCA1 gene, more than 500 alleles are known worldwide. Even though only 4 are seen among the French-Canadian, several inborn errors of metabolism are also more common in this group. The French-Canadian have ideal characteristics for gene discovery many generations since founding. Few founders, around about 2,500, a high rate of population expansion percent increase per generation, a large present day populations, that is about 6 million, and most marriages within the group. Founder effect in human population. While discussing about the founder effect in human population, the French-Canadian population exemplifies genetic drift because the people have kept mostly to themselves within a larger population. The French founded Quebec city in 1608. Until 1660, the population grew as migrants arrived from France and then began to increase from birth. More than 10,000 French has arrived by the time the British took over in 1759, but many of them had westward. In Quebec, religious, language, and other cultural differences separated the French and English gene pool. The French-Canadian population of Quebec grew from the to 2,500 or so. So overall, the basic idea behind this is that the people homozygous, we can say that the people homozygous for the allele have achromatopsia, that is complete color blindness, extreme light sensitivity, and overall idea this particular effect is complete color blindness, extreme light sensitivity, and poor visual acuity. Achromatopsia is rare in most population. This is about less than 1 in 20,000 population. And among the 3,000 Pingley-Lapsis Islander, the frequency is 1 in 20. This is the whole idea behind this. Founder effect in human population. It has been seen that the higher frequency of allele for achromatopsia is not due to a selective advantage. It is just a result of chance. So founder effect followed by further genetic drift resulted in current high frequency. Or we can simply, if we relate with the study, founder effect can be studied at the phenotype and the genotype level. Phenotypically, founder effect is indicated when a community of people known from local history to have descended from a few founders have inherited traits and illness that are rare elsewhere. This is striking among the old order Amish and Mennonites of Lancaster County, Pennsylvania. Often worried parents would bring their ill children to medical facilities in Philadelphia. Over the years, researchers realized that these people are subject to an array of extreme rare conditions. For example, Victor, founder of online Mendelian inheritance in man, discovered and described cartilage hair hypoplasia after 6 Amish children died at a Philadelphia hospital from chickenpox in 1965. Part of their inherited syndromes was impaired immunity. Unlike McCormick's made the connection, the other symptoms, including dwarfism, spasm hair, and anemia, were not recognized as part of a syndrome. Today, as many genetics study inherited disease common among the Amish and Mennonites, treatments are becoming available from spatial diet to the counter inborn error of metabolism to the gene therapy. In addition to the historical record, differences in allele frequency in a small population compared to those in the general population suggest a founder effect. The incidence of certain disease in Lancaster County, for example, in astounding maple syrup, urine disease effect 1 in 225,000 newborns in the United States, but 1 in 400 newborns among the Lancaster family. A research fellow at a children's hospital in Philadelphia discovered that cerebral palsy in several young children from Lancaster County attributed to oxygen deprivation at birth was actually an inborn error of metabolism, and he called it as a glutaric aciduria type 1. He went from farm to farm, tackling case against genealogical record, and found that every family that could trace its root back to the founder had members who had the disease. Today, in 1 in 200 newborn in this population have the condition. A mutation that is the same in all affected individuals in a population is strong evidence of founder effect due to descent from shared ancestor. The Bulgarian gypsies who had a galactokinase deficiency, for example, all had a mutation, but in extremely rare elsewhere. In contrast, a population with several mutations that causes the same disorder is more likely to have picked up those variations from people joining the group. rather than from descent from shared founder. Very often, when a disease-associated allele is identical in a DNA sequence among people in the same population, so is the DNA surrounding the gene. This pattern indicates that a portion of a chromosome, rather than just the disease-causing gene, has been passing among the members of the population from its founder. For this reason, many studies that trace founder effect examine haplotypes that include tightly linked genes. When historical or genealogical records are particularly well kept or recent, founder effects are sometimes be traced to the very beginning. This is the case for the population of South Africa called as Afrikaner. The 2.5 million Afrikaners descended from a small group of Dutch, French and German immigrants who had huge families, often with as many as 10 children. In the 19th century, some Afrikaners migrated northward to the Transvaal province, where they lived in isolated until the Boer War in 1902 introduced better transportation. Today, 30,000 Afrikaners have porphyria variegata. All affected people descended from one couple who came from the Netherlands in 1688. Today's allele frequency in South Africa is far higher than that of Netherlands because the founding couple had many children who in turn had large family, passing on and amplifying the dominant mutation. Founder effects are also evident in most common illnesses where population had different mutation in the same gene. BRCA1 breast cancer, for example, is most prevalent among Ashkenazi Jewish people. Nearly all affected individuals have the same 3 base deletion. In contrast, BRCA1 breast cancer is rare in black, but it affects families from the Ivory Coast in Africa, the Bahamas, and the southeastern United States. They share a 10 base deletion, probably inherited from West Africa ancestral to all 3 modern groups. Savages brought the disease to the United States and the Bahamas between 1619 and 1808, but some of their relatives who stayed in Africa have perpetuated the mutant allele there. Now let's discuss about bottleneck effect. A population bottleneck occurs when many members of a group die and only a few are left to replenish the number. The new population has only those alleles in the small group that survived the catastrophe. An allele in the remnant populations might become more common in the replenished population than it was in the original larger group. Therefore, the new population has a much more restricted gene pool than the larger ancestral population, with some variant amplified, other diminished. Population bottlenecks can occur when people or other animal colonized island. An extreme example is seen among the Lapis people of the Eastern Caroline Island in Micronesia. 4 to 10% are born with Pinglapsi blindness. It is an autosomal recessive combination of color blindness, nearsightedness and cataract, also called as achondroplasia. So, we can say that bottleneck effect is a sudden reduction in population size due to a change in the environment. A new gene pool may not reflect original. If the population remain small, it may be further affected by genetic drift. A population bottleneck occur when the size of genetically diverse population falls, remain at this level for a time and then expand again. So the new population loses some genetic diversity if alleles are lost. Example of a population bottlenecks. A population bottlenecks occur when many members of a group die and only a few are left to replenish the number. The new population has only those alleles in the small group that survive the catastrophe. An allele in the remnant population might become more common in the replenished population than it was in the original larger group. Therefore, the new population has a much more restricted gene pool than the larger ancestral population, with some variant amplified, others diminished. Population bottleneck can occur when people or other animals colonize island. An extreme example is seen among the Pingley-Lapses people of the Eastern Caroline Island in Micronesia. 4 to 10% are born with Pingley-Lapse blindness, and which is an autosomal recessive combination of color blindness, nearsightedness, and cataracts, are also achromatopsia. Elsewhere, only 1 in 20,000 to 50,000 people inherit the condition. Nearly 30% of the Pingley lapses are carrier. The prevalence of the blindness among the Pingley lapses stemmed from a typhoon in 1780 that killed all but 9 male and 10 female who founded the present population. This severe population bottleneck plus geographic and cultural isolation increased the frequency of the blindness gene as the population resurged. A more widespread population bottleneck occurred as a consequence of the early human expansion from Africa discussed in earlier chapter. As number dwindled during the journey and then were replenished as people settled down, mating among relatives led over time to an increase in homozygous recessive genotype compared to ancestral populations that maintained their genetic diversity in Africa. These bottlenecks are reflected today in the persistence of genetic diversity among African populations. The lack of genetic diversity in some modern human population is evident as runs of homozygosity, which are chromosome regions that vary little from person to person. Runs of homozygosity generally represent regions that are inherited from shared ancestors. They are common, for example, in highly purebred dogs. So the figure 1 explain the population bottleneck. A population bottleneck occurs when the size of a genetically diverse population falls, remains at this level for a time and then expand again. The new population loses some genetic diversity if alleles are lost. This figure explains the changes or the process of bottleneck effect, or we can say that it illustrates schematically the dwindling genetic diversity that results from a population bottleneck. For example, today's cheetah live in just two isolated populations of a few thousand animals in South and East Africa. Their numbers once exceeded 10,000. The South African cheetahs are so alike genetically that even unrelated animals can accept skin graft from each other. Researchers attribute the cheetah's genetic uniformity to two bottlenecks. one that occur at the end of the most recent ice age when habitat changed, and other following mass slaughter by humans in the 19th century. However, the good health of the animals today indicates that the genes that have survived enable the cheetah to thrive in their environment. This figure explains the genetic drift and loss of genetic variations. Here in this two figure, you will see the changes where on one side see the range of genetic parasiticin, then their distributions, their changes in the form of figure as a pre-bottleneck regions during 1820 and post-bottleneck region during 1993. A great drastic changes that we have seen. Similarly, the post-bottleneck in this area, it shows that how the variational changes take place according to time. So from this figure, we will clearly find out number of allele per locus that undergo changes and percentage of each hatched that undergo some changes based on the phenomena of genetic drift and the loss of genetic variation. Okay, now let's try to conclude the whole modules with some important points as a summary. Genetic drift produces steady decline in heterozygosity. Frequency of heterozygosities is highest at intermediate allele frequency. As one allele drift to fixation, the number of heterozygotes inevitably decline. It is significant in small population, that is genetic drift. It causes allele frequency to change at random. It is also genetic drift. Genetic drift can lead to a loss of genetic variation within the population. It can cause harmful allele to become fixed. Population follows unique path. Genetic drift has strongest effect on a small population. Given enough time, even in a large population, genetic drift can have an effect. Genetic drift leads to fixation or loss of allele, which increases homozygosity and reduces heterozygosity. Thank you.